Hello, my name is Mr. Pittenger, and welcome to demonstrating the cell cycle mitosis with pipe cleaners. Today we are first going to look at the materials that are within your pipe cleaner kit. Within each kit you should have a total of 4 of these small yellow pieces, 2 large blue pieces, 2 large red pieces, 2 small blue pieces, 2 small red pieces, along with 4 of the white beads. You will place those white beads on the chromosomes as you see here. To begin, we will not need all of the parts within our kit. We will only need the blue and the red pieces that have beads on them for now. And then I will only need 2 of these yellow pieces to begin. So now we are ready to begin our demonstration of the cell cycle or mitosis. We are starting out with a single cell, so one cell. And this cell is a body cell, also known as a somatic cell. And within our somatic cell, or body cell, we are going to take a look at what pieces we have, starting with the blue pieces. These 2 blue pieces, the big blue and the small blue, are representing what is known as paternal chromosomes. These are chromosomes that this organism inherited from its father. And where did these chromosomes come from? They came from this organism's father's sperm cell. The other 2 chromosomes that we have colored in red, the big red and the small red, these are known as maternal chromosomes. These are chromosomes that this organism inherited from its mother. And where did these chromosomes come from? They came from the mother's egg cell. So both of these, the father's sperm and the mother's egg cell, are known as haploid cells because they contain half the genetic information as a body cell. So in this case, these are haploid sex cells or gametes that each contain 2 chromosomes. And what happens during fertilization is the father's sperm carrying these 2 blue chromosomes fertilizes the mother's egg carrying the 2 red chromosomes to form our first cell that all of us begin as, known as a zygote. Looking at these chromosomes, each one of these pipe cleaners is representing a chromosome within our cell. Each chromosome is made up of a long DNA molecule that is wrapped in protein. And these chromosomes that we have in our cell model are found in the center region of the cell, known as the nucleus of the cell. So for our cell model here, we have a total of 4 chromosomes present. And the order in which they are placed within this nucleus does not matter. We are going to begin our cell cycle in this phase known as the growing phase. It's known as interphase. And really within interphase, these chromosomes are not visible. They are tangled up in this string-like form that looks like spaghetti. And this DNA is called chromatin. Before we proceed, we are going to identify our 2 small yellow pieces over here. These yellow pieces are cell organelles known as the centrioles of the cell. So we're beginning in interphase. And interphase can be broken up into 3 substages. The first substage of interphase is called G1 or growth 1. During this time, the cell, which is our background, the table, the cell is getting larger in size. It is preparing for cell division, and all of the cell organelles double in number. In our kit, the only cell organelles that we have present are the centrioles over here in the yellow. So to show this, we are going to show that these centrioles double in number. So they will double from 2 to 4. So during G1 of interphase or growth 1, the cell grows in size, and the centrioles, along with all the other organelles, double in number. But what happens to a cell if it grows too large? Well, the cell has really 3 options. The cell can stop growing, it can choose to divide, or if the cell does not choose either of those 2 fates, the cell could end up dying. The cells that are best at supplying nutrients, expelling waste, and communicating are the cells that are small in size. And this surface area to volume ratio is important for cells. It does change as a cell increases in size. The surface area to volume ratio decreases as the cell increases in size because the volume increases at a much faster rate than the surface area. So that is a bad thing for cells. What is the difference between the surface area and volume in relation to cells? The surface area includes the number of pathways or doors for materials to enter and exit the cell, whereas the volume is the distance that those materials have to travel either to get in or out of the cell. So the most efficient cells will have a high surface area and a low volume. That's what cells want in order to be efficient. So after G1 of interphase, the cell next enters the second part of interphase that is called the S phase or synthesis phase. So during the S phase of interphase, DNA replication occurs. So each of these chromosomes, which are not visible yet, they're still in this form called chromatin will make a copy of itself. That process is called DNA replication, so it looks like this. We still have the same number of chromosomes in the cell. We still have 4 chromosomes, but each one of these chromosomes is made up of identical parts or identical halves that are formed after DNA replication, and these identical parts of the chromosome or identical halves are known as sister chromatids. So the left and right pieces of this chromosome are called sister chromatids because they're identical. This one just made a copy of itself during DNA replication. The same is true for my other 4 chromosomes here. So we have 4 chromosomes, but each chromosome now is made up of 2 identical parts that we call sister chromatids. These sister chromatids are held together by these white beads, and each white bead is called the centromere of the chromosome. The word centromere means center part. So this DNA replication occurs during our second part of interphase called S, or synthesis. The term used to describe a pair of chromosomes, such as our large blue and large red chromosomes, that all have the same traits. The term that we use to describe these are homologous chromosomes. So here is one pair of homologous chromosomes, my big blue and my big red. And then also I have another pair, my small blue and my small red chromosomes are also homologous to each other. Homologous means they have all of the same kinds of traits, and we can normally identify them by size. So same size but just different color with the pipe cleaners. So I have a total of 2 pairs of homologous chromosomes in my cell after DNA replication occurs. The third and final part to interphase, or the growing phase, is called G2, or growth 2. And during this time, the cell continues to grow and protein synthesis occurs. So all of these events that I have showed to you occur during interphase, the longest part of the cell cycle. The cell cycle is regulated by these groups of proteins that are called cyclins and CDK enzymes, which stands for cyclin-dependent kinases. So these are the regulators of the cell cycle. They set the pace for which all of these phases occur throughout cell division. If a cell becomes damaged and cannot repair itself, then these cyclins and CDK enzymes will trigger this cell to commit programmed cell death. or cell suicide, which is called apoptosis. Apoptosis is a mechanism that keeps damaged cells from dividing and making more copies. What could happen to the cell if the cell cycle is not controlled by these cyclins and CDK enzymes? Then the cell could become cancerous. Cancer is uncontrolled cell growth and division of cells. So how are cancer cells different from normal cells? These cancer cells spend less time in the phases of the cell cycle, mainly interphase. So they will spend a lot less time here in this first part of the cell cycle known as interphase. Really, throughout interphase, these chromosomes will be in this tangled form that we call chromatin, but they have been separated in order to show you the events. So that completes the first main stage of our cell cycle, called interphase. The name of our second main stage of the cell cycle, in which the cell's nuclear material is actively dividing, is called mitosis. And the first stage of mitosis is called prophase. Pro meaning before or first. And a lot of events happen during prophase of the cell cycle. So during prophase, these centrioles working in pairs are going to begin to move toward opposite poles of the cell. And by the end of prophase, they will be there. And these centrioles line up at right angles. They're perpendicular to one another. So they will be at opposite poles of the cell. Coming out of these centrioles will be these protein fibers that are like protein ropes that we call the spindle apparatus or spindle fibers. Also during prophase, the nucleus and nucleolus will disappear. And this DNA in the form of chromatin will start to condense into visible chromosomes that can be seen under the microscope. So we can see during prophase that there are 4 chromosomes. The second phase of mitosis that comes after prophase is called metaphase. And during metaphase, these chromosomes will randomly line up down the center of the cell. Like this. So the order in which you line them up does not matter as long as they are in a straight line. So in metaphase, these chromosomes form a straight line. They meet in the middle or the equator of the cell. I call this the marriage phase of mitosis. They're together down the center. They have been moved by these spindle fibers, which are now attached to the center part of the chromosome, the bead called the centromere. So these spindle fibers have attached and moved these chromosomes to the center of the cell. Following metaphase, the next part of the cell cycle is called anaphase. So during anaphase, these sister chromatids are pulled apart and move in opposite directions. Just like this. And it's okay if you just leave the beads on those 4 chromatids, that is fine. So notice how these sister chromatids are separated from one another. During anaphase, they come apart. So remember, during anaphase, think apart. It's also referred to as the divorce phase because these sister chromatids are separating. They are never, ever, ever getting back together, as Taylor Swift would say. So then in the 4th and final phase of mitosis, the cell reaches telophase. During telophase, these sister chromatids reach the poles, so they arrive at their final destination. And the nuclei, which is plural for nucleus, will reform around those chromosomes at each end of the cell. Same thing over here. And the chromosomes return back to chromatin, just like they were in interphase of the cell cycle. So these will become string-like and look like spaghetti once again. And we call the DNA chromatin when it's in this form. Same thing on this side of the cell. So really, during this time, this cell is starting to begin to look like 2 cells, but it has not quite split yet. And that happens in the 3rd and final part of the cell cycle. So the cytoplasm will divide in this last part called cytokinesis. So during cytokinesis, the cell will split down the middle, the cytoplasm will divide, and we now have 2 daughter cells. They are both body cells, and each cell is a diploid cell, meaning that it has 2 full sets of chromosomes. So these are both diploid cells, each containing the same genetic material. Each one has 4 chromosomes if we were to pull this chromatin apart. Each one has a big blue, a big red, a small blue, and a small red, just like our initial starting cell. If these were human body cells, then each human body cell would have 46 chromosomes. The cell cycle was a very long process, and the cell spends most of its life in that very first part that we've talked about, known as interphase. So the cell will spend about 90% of its life, unless it's a cancer cell, in interphase, and that is known as the growing phase of the cell cycle. So now our cell cycle process is complete. We have made our two body or somatic cells, each having 4 chromosomes in this model. And what are 2 reasons why mitosis is important to living organisms? Mitosis is important for cell growth and repair of living things. Thank you for watching. Good luck.